Microscopy helps people figure out tiny words. A 10 micrometer cell can be magnified many times and becomes observable. However, most cells are transparent, so cameras and human eyes cannot easily see them without staining. But some stains are time-consuming and might kill the cell, making live cells hard to see. Recently, some label-free image techniques are developed. One of them is quantitative phase imaging. How does it work? Light travels slower in cells than in water, and causes delay in phase, which is proportional to cell mass. Quantitative phase imaging can record phase delay and turn into image. One can know both cell shapes and information inside cells by this means. Our labs worked on developing tools helping biological research with quantitative phase imaging. The project I engaged aimed to create an automatic label-free system distinguish dead cells from live cells and identifying two types of cell death, apoptosis and necrosis. Apoptosis is also known as programmed cell death. Some inner part of the cell plans and tells the cell how to die. By contrast, necrosis is a non-programmed cell death. It happens because of external factors, such as toxin. Cell death distinguishment is widely used in biological research, such as cell culture monitoring and drug development. In the test of the candidate of new drugs, researchers usually treat cells with the candidates and observe whether they die or not to confirm this candidate to be not harmful. And dead cell classification can help drug modification. When cell death occurs, some features change. For example, apoptosis cells tend to shrink and become smaller. Necrosis cells tend to swell and become larger. To build up a cell death identification system, we first found out which and how cell features change in quantitative phase images, and used this information to train computer model. Finally, the trained model is expected to identify dead cells in new images. Label-free dead cell identification may help cell research go on smoothly by providing long-term detection of cell death without staining effects.